Have you ever wondered why does the diverse kind of food, such as bread, meat, nuts, can be resolved after they are sent to the digestive system in our body and becomes nutrition that sustain our lives? Or have you ever wondered why do the dirty clothes become new and clean after they are washed with the laundry detergent? Enzyme is the key that makes this magic happen. In this video, I'm going to introduce you to the magic world of enzyme and do a biological experiment through which you will learn what factors influence enzyme functions, how to measure enzymatic activity, and how to utilize the characteristics of enzyme to the best. First, let us learn about what is enzyme. It is a catalyst for chemical reactions. The appearance of it accelerated the chemical reaction, but itself is not a part of the reaction, so its amount will stay the same during the reaction and can be reused after the reaction. Enzyme is mostly made of protein. Some of them are RNA. It has a specific shape that enables it to bind with a specific substance that fits. That substance is called a substrate. When a substrate is combined with the enzyme and form a complex, the enzyme would change its shape so that the substrate would break into other kinds of substances or combine with another kind of substrate, resulting in products. Because enzyme is a very efficient catalyst which can increase the reaction rate with remarkable power by at least a millionfold, humans learn to utilize its efficiency by extracting it and put it into mass productions for diverse use, such as medicine. For example, there is an enzyme called lactase, which can catalyze the hydrolysis of beta-glycosidic bond in the disaccharide lactose, releasing galactose and glucose. This enzyme is highly expressed in human body since newborn babies need to digest the breast milk to generate energy. However, there are a group of people who have congenital lactase deficiency, or CLD in short, who lack the ability to express lactase as normal people. A more common term to describe their symptoms is lactose intolerance. This will result in difficulty in feeding them as babies, and will also deprive them of the opportunity to the variety of delicious food containing lactose. Lactase tablet is a product that can benefit people with lactose intolerance. This experiment is going to figure out how does time factor influence the enzymatic activity of lactase, so that the patients who take this medicine can control the time between the intake of medicine and the lactose-contained food. To measure the enzymatic activity, we can measure the concentration of the product. The more the product, the more the enzymatic activity. The method we use to measure the concentration of the product is called spectrophotometry. This equipment is called spectrophotometer. It can scan the sample we put in with a 96 well plate on the light wavelength that we set. When we put in the plate and let a spectrophotometer scan it, it can tell the concentration of the solution by measuring the sample's light absorption. Bigger the absorption, bigger the concentration. Therefore, for a spectrophotometer to work well in measuring the concentration, being able to absorb light is the key. However, none of lactase hydrolysis products absorb light. So, a chemical named OMPG is used in this experiment as the substrate in substitution of lactose, for it also possesses the beta-glycosidic bond that can be hydrolyzed by lactase and that its products can absorb light. The experiment process is followed. Firstly, prepare a commercial lactase tablet. Crash it into powders to extract the lactase. Secondly, prepare 14 microcentrifuge tubes. Label 2 of them as control A and B. Label the rest from tube 1 to tube 12, and divide them into 4 groups with 3 in each group, so that we can do the experiment in 4 different time conditions. Prepare 3 contents to be put into the tubes: PBS buffer, substrate, and enzyme. No substrate in control A, no enzyme in control B. For the rest, add buffer and substrate in each. Then, set the dry bath to 37 degrees Celsius. Add enzyme into tube 1, and immediately put the tube into the dry bath. After 30 seconds, immediately add stop solution to stop the reaction. Repeat the process for the rest tubes. Change the reaction time to 60 seconds, 90 seconds, 120 seconds. 3 tubes for one time length. Lastly, pipette the content from each tube to the wells of a 96 well plate and measure the absorbance at 450 mm, for this is the absorbance maximum of the product. Analyze the data, plot the standard curve. From the curve that we drew from the result, we can see that the concentration of product increased with the increase of time. Therefore, the lactase enzymatic activity is proportional to time. With the knowledge of how lactase enzymatic activity changes through time, the people with lactose intolerance can understand the mechanisms of the medicine they take better and control the time between when they take the medicine and when they eat food. We are approaching the end. Before we end this video, I want to express my thanks to all the people that have helped me in this session of biology's journey. Firstly, I want to express my thanks to Professor Lee, who has encouraged me and answered all my concerns and questions when I got back scores in exam. And he always delivers interesting, informative lectures. Thank you. Secondly, I want to thank Tutor Lu for grading my homeworks patiently and generously, and he always gives me timely and useful feedbacks that helps me to improve in the future. And of course, I want to thank the members of my Think Pair Share groups. They are MJ, Yanjia, and He Yu. Thank you. It was because of your discussions that helped me to learn biology better. Thank you all. Hope to see you again in the future academic journey. Bye!